So hi, my name's Ian Battersby. I'm one of the clinicians at Davies Veterinary Specialist and I work in the internal medicine department. And welcome to the second of our webinar series on antibiotic prescribing. Today we're going to just review and reflect a few points on how our antibiotic prescribing may influence the development of resistance bacteria. So first of all, I think it's always good just to review a little bit about how bacteria resist the influence of antibiotics, and we're just going to do that briefly now. So I'm sure we'll hopefully all remember from our microbiology days there's a number of different ways that bacteria can acquire resistance mechanisms to antibiotics. They also have natural resistance, so if you, for example, don't have if a bacteria doesn't have a cell wall, such as a mycoplasma, then an antibiotic that is targeting the cell wall, such as a penicillin, which is a beta-lactam, that's going to have no effect on that bacteria. So that's natural resistance, but we also have acquired resistance. So this is the acquisition of pumps and mechanisms to expel the antibiotic. And bacteria can do that in a number of ways. First of all, they can mutate spontaneously. Secondly, they can acquire DNA from other bacteria in the form of plasmids, or the mobile DNA elements that move, the little circular elements move from one bacterial species to another. And then there are also viruses and bacteriophages that can make this all quite a complex scenario. How do bacteria reduce their susceptibility to the antibiotics? Well, I've used the word susceptibility because all of these mechanisms potentially have a point where they're going to be overrun, but it's just clinically we can't achieve those concentrations. So, bacteria will either alter the target of the antibiotic, they'll reduce drug entry or excretion, as I mentioned, pumps before. They'll work out different ways to metabolize the drug, so beta-lactamases are one mechanism that we're most familiar with. Or they may produce alternative metabolic pathways to bypass the area where the antibiotic targets. And I think it's important that we do just refresh, the difference between in vitro and in vivo resistance. So, we get very much focused on the resistance in the plate, but actually as clinicians it's important that we focus on whether we're resolving the infection in the patient. And it's not just the use of the antibiotic in those patients, it's not just the antibiotic resistance profile that's important. There's a number of other factors, and it's important that we bear that in mind when we're considering treating these more difficult bacterial infections. So when we consider our clinical resistant infection, there's a number of different factors that we need to consider. So first of all, there's individual bacterial factors. There's the pharmacodynamics of the drug, and then finally there's the host factors. So if we cover bacterial factors first, well, we've already mentioned it, we've got the individual sensitivity of the bacteria to a certain antibiotic, and can we achieve that level of antibiotic concentration in our target area? And I guess that influences and touches on some of the pharmacokinetics. We've also got the individual virulence factors as well for the bacteria. Resistance doesn't necessarily mean it's a more virulent strain, but if it does cause an infection, it can obviously be a lot more problematic to treat. Pharmacokinetics is the second area, and will the antibiotic reach the target area? So this can be reliant on the acidity, influenced by the acidity, pH of the area, the blood-brain barriers and prostate barriers, the lipogenicity of the drug all play a role. And then finally we've got our individual host factors, and that can be split into two broad areas. First of all, you've got the blood supply, which influences how the drug can get to the target area, whether there's necrotic tissue, whether that abscess, for example, is draining well. And then we've also got the immune system of the patient. So if you've got antibiotics don't replace your immune system in resolving an infection, they assist your immune system in fighting an infection. So if you are immunocompromised, it's obviously more challenging to resolve that infection than in a fitter, healthier individual. So now we're just going to talk a little bit about how our prescribing of antibiotics can influence the evolution and development of resistant bacteria. So I'm just going to do that with a fictional example, and this diagram here shows a fictional kind of bacterial population that's mixed, and each colour denotes a different species. And we've decided that we're going to eliminate, we've got an infection and we need to eliminate some of those bacteria. And to do that, we're going to use an antibiotic. And the bacteria that we're interested in are the ones denoted with a red X. And we prescribe an antibiotic, which is this big red X as well. And this is a broad spectrum antibiotic. So this is an antibiotic that will target the bacteria we're interested in, but it's broad spectrum, so it kills lots of, or it has an effect on lots of different bacteria. And as you can see, as we move the animation forward, we're starting to eliminate the bacteria we're interested in, but we're also beginning to eliminate the commensals that we're not interested in. However, there is one bacteria on that slide that is has a resistance mechanism to the antibiotic we're using, and that's the black one. Now, on its own, that resistance doesn't hold any survival advantage. In fact, the other bacteria in this slide are better at surviving in that environment. They've got other mechanisms of acquiring nutrition. But we put the antibiotic in place, and all of a sudden that antibiotic resistance mechanism becomes a survival advantage. And as we move on through the animation, because there's less bacteria around to compete in the environment, that resistant bacteria now has a survival advantage and then can propagate. So the best analogy you can use really is Darwinian selection of the fittest. A survival advantage that may not hold any advantage until a selection pressure is put in place. So by using an antibiotic, we understandably want to treat the infection, but if we use it irresponsibly, use it for a very prolonged period, we put a positive selection pressure for resistance. And this is just some data from, it's very, one of our ophthalmologists, David Gould, put together. He did a quick survey of some of the conjunctival swabs of dogs that have been referred for conjunctivitis. And it's just interesting to note, I think this is a population of maybe about 30, 40 dogs, that if you look at the fusidic acid sensitivity, quite a lot of those bacteria are resistant to fusidic acid in that bacterial, in that population of dogs, which is a very commonly prescribed antibiotic for conjunctivitis. But when we talk about resistance, it's not, resistance, using one antibiotic doesn't necessarily mean that you will only be resistant to one antibiotic that you're using. These bugs can have DNA that codes for resistance to multiple antibiotics, and that may be because the plasmid contains DNA for multiple mechanisms of resistance, or it may be that the bacteria contains multiple plasmids. So that's just a review of the traditional model for selection of resistant bacteria. But if you look at the culture profiles in a lot of the laboratories, those resistance profiles don't often influence, don't correlate with that all the time. So there are other mechanisms by which our antibiotic prescribing are potentially influencing the development of mutations and resistance. Of course, there'll be non-antibacterial mechanisms as well, but one I want to mention is the hypermutable state. So this is when an antibiotic concentration is insufficient to kill a bacteria, but it is having an effect at the intracellular mechanisms. And some of those mechanisms then don't work as well. So whenever a cell reproduces, DNA is replicated, but the complexities of that process mean that there are obviously going to be errors in the coding. We all have mechanisms that minesweep kind of correct those errors, but in the hypermutable state, the antibiotics influence those gene coding correction mechanisms, so mutations are more likely to happen. And sometimes it's suggested that bacteria can be up to 200 times more likely to develop a mutation in subtherapeutic concentrations of antibiotics. Now, that doesn't necessarily mean that that mutation will result in an antibiotic resistance mechanism. It might be that that mutation is lethal for that bacteria. It might be that that mutation does nothing at all. But obviously, the more times a bacteria mutates, the more likely you are to get mechanisms developing. And a really nice video to watch, and I put the reference here and we'll include it on the post, is available on YouTube. And it's a video just highlighting the evolution of bacterial resistance to antibiotics on a giant Petri plate. And I don't want to spoil the ending, but even people who aren't geeky about this kind of stuff like me have found it very interesting. It's only a short video, it only lasts about 2 or 3 minutes, but I'd strongly advise you, if you can, just have a quick review on YouTube. So I guess in summary, when we consider our antibiotic prescribing, if we're using them too often, if we're using them for too long a period, we could influence selection of resistant bacteria. If we underdose and don't hit therapeutic levels so we can treat effectively, then also that's going to potentially influence the development of resistant bacteria. So thanks again for listening to this short webinar. We'll be moving through the series, and I hope you'll be able to tune in for the next webinar, where I'll be discussing how we reflect on whether antibiotics are indicated in certain clinical situations. And if you'd like to follow us on Facebook, LinkedIn or Twitter, they'll be able to give you more information about when those webinars will be released.